Hello students, welcome to EPG Pathshala. I am Dr. Ranjana Saxena, Associate Professor in Zoology from Dayal Singh College, University of Delhi. Today we are going to discuss on the module Pathogenicity, Diagnosis and Prophylaxis of Entamoeba histolytica from the paper Parasitology. Please recall students, we have done the morphology and life cycle of Entamoeba histolytica in our previous module. After studying the unit, you will be able to understand the medical importance of Entamoeba and explain the invasive and non-invasive infection caused by Entamoeba histolytica. Highlight the symptoms of the disease caused by Entamoeba histolytica. You will also be able to tell the diagnostic features of amoebiasis and suggest various methods for the prevention and control of the parasite. Now let us start this module with pathogenesis of Entamoeba histolytica. Pathogenesis depends upon three factors: parasite virulence, host resistance, and condition of the intestinal tract. Now talking about the parasite virulence, the infection can either be non-invasive or it can be invasive. The non-invasive infection is localized and confined to the lumen of the large intestine and is usually seen in people with normal immune system. As the name suggests, in the invasive infection, the trophozoites can penetrate the intestinal wall and cause lesions. This can be normally found in persons with a weak immune system. It is the virulent form that invades the intestinal tissues and cause disease. Certain strains of Entamoeba histolytica can be more virulent than the others. Entamoeba histolytica produces 4 virulent factors. These are amoebic lectin, ionophore-like protein, toxins and hemolysins, and hydrolytic enzymes. Now let us talk about the second factor, which is host resistance. The factors that influence the course of action for the host resistance to be effective is immunity, immunosuppressive drugs, corticosteroid therapy, nutrition, alcoholism. Talking about the third factor, which is condition of the intestinal tract. Now, condition of the intestinal tract depends upon the bacteria and the intestinal flora. Invasion of amoebic trophozoites can be divided into 4 stages. The first is colonization of the large intestine. Second, adherence of Entamoeba to the colon mucosa and lysis of the mucosa cells. Third, invasion of intestinal mucosa. And the fourth is extra-intestinal invasion of tissues. Colonization of the large intestine. Let us take one by one all the 4 factors. Colonization of the large intestine. This is the first stage of amoebiasis. The Entamoeba come in contact with the mucosa and submucosa of the colon and remain there for a long time, thus facilitating the invasion. These Entamoeba facilitate the secretion of mucus, which makes the epithelial cells of the colon more susceptible to invasion by them. The presence of bacteria and host digestive food fulfills the necessary metabolic requirement of these amoebic trophozoites. The next factor is adherence of Entamoeba to the colon mucosa and lysis of the mucosal cells. The trophozoites now adhere to the mucins of the colon, epithelial cells and leukocytes. Adherence is mediated by galactose-specific lectin. Lectin is a heterodimeric glycoprotein composed of 2 subunits, 170 subunit and 35 subunit. The 35 light subunit is covalently attached to the heavy 170 subunit through disulfide linkages. Each lectin subunit is represented by multiple genes within the Entamoeba histolytica genome. The 170 one is rich in cysteine and is responsible for adherence of trophozoites to galactose receptors present in the intestinal mucosa. This protects the entamoeba from the action of complement. The components of the complement are prevented from being deposited onto its surface. The participation of light subunit in lectin function is not really known to us. After adherence, trophozoites lyse the mucosal cells, which are the target cells, by the ionophore-like protein. Now, invasion of the intestinal mucosa by the Entamoeba histolytica, let us talk about it. Cysteine proteinase, cysteine kinase you can call it, is an extracellular enzyme produced by Entamoebae which causes proteolytic destruction of the tissue. The enzyme causes tissue destruction by digesting the extracellular matrix and also by breaking down IgA molecules and reducing certain components of the complement. A polymorphonuclear response of the host is also responsible for the destruction of the host tissues. Now, invasion of the colon and cecum actually takes place at the intergranular site of the intestinal mucosa, as the site is of low resistance for the trophozoite. Tissue destruction caused by Entamoeba gives rise to flask-shaped amoebic ulcers. Now we are going to do the extra-intestinal invasion of tissues, which is one of the factors. From the intestine, the Entamoeba are carried by the portal blood circulation first to the liver and then from there to the various organs like lungs, spleen, heart and brain. They are, however, not carried to these organs directly from the intestine. The pathogenic strains of Entamoeba is resistant to the complement-mediated lysis in the bloodstream and other organs that they invade. This is probably because of the activity of cysteine proteinase that it produces. This enzyme inactivates the complement factor C3, thus limiting the host inflammatory response. Let us talk about the symptomatic amoebiasis. Symptomatic amoebiasis can be intestinal or extra-intestinal. Intestinal one includes non-dysenteric amoebic colitis, amoebic dysentery, chronic amoebic dysentery, and amoebic colitis, whereas the extra-intestinal one means that they invade the liver, the lung, the spleen, and the brain. Pathology of the intestinal lesion, if we talk about it, it causes amoebic ulcer in the large intestine. Some of the trophozoites, what they do is they penetrate the columnar epithelial cells of the mucosa in the colon, causing ulcers. E. histolytica, that is Entamoeba histolytica we are talking about, secretes a proteolytic enzyme, histolysin, which not only digests the host epithelial cells, causing necrosis of the tissue, resulting in ulcer, but also facilitates the penetration of the trophozoites into the mucosal layer. However, the ulcers remain confined in the colon, being most numerous in the cecum, followed by sigmoidorectal region. The ulcers contain active, rapidly dividing trophozoites and is covered by mucus. However, the intervening mucosa between the ulcer is usually healthy. The ulcer in the mucosa and submucosa measure from a pinhead to an inch in diameter. Entamoeba histolytica may cause amoebic appendicitis and amoebomas. Amoeboma is a tumor-like inflammatory mass of colon. Necrosis, inflammation, and edema of mucosa and submucosa of colon, together with large number of trophozoites, can be seen. Clinically, the symptoms are usually acute, with abdominal pain, appreciable mass, and dysentery. Talking about the symptomatic intestinal amoebiasis, it may progress from mild diarrhea to severe hemorrhagic dysentery. Uncomplicated, simple amoebiasis, what we call as non-dysenteric amoebic colitis. Now, what is that? The symptoms of this include intermittent diarrhea, mucus, cramps, and flatulence. Flatulence means excessive gas in the alimentary canal. Patients have small amoebic ulcers with less colonic inflammation. It is usually chronic. Then acute amoebic dysentery can also be caused by amoebic Entamoeba histolytica. Now, it is the invasive intestinal amoebiasis and occurs when the parasite invades the intestinal wall and ulcers develop. The stool of patient contains blood and mucus. A large number of trophozoites are also discharged with the slough in the feces. Patients have severe abdominal pain. The third type is chronic amoebic dysentery or amoebic colitis. This has a high mortality rate and tends to occur in malnourished people, people taking corticosteroids, and people with compromised immune system are basically suffering from chronic amoebic dysentery or amoebic colitis. It is characterized by intermittent diarrhea over a long period of time. Patients have severe abdominal pain, colon perforation, and hemorrhage frequently occurs. Often, it is misdiagnosed as ulcerative colitis or irritable bowel syndrome. Now, talking about the extraintestinal sites. Extraintestinal means outside the intestine. The trophozoites are motile and actively they move about. Sometimes they may find their way from the colon into the radicals of the portal vein and then get carried to the extraintestinal sites like the liver, the spleen, lung, or maybe the brain also. Clinical manifestations of the extraintestinal amoebiasis depends on the organ which is being infected. Now, amoebic liver abscess may be caused by them. It is the most common form of extraintestinal amoebiasis, that is the amoebic liver abscess. It is the most common form of extraintestinal amoebiasis. The trophozoites are carried from the ulcers in the colon and cecum to the right lobe of the liver by the right branch of the portal vein. Here they multiply and cause liver abscess. The condition occurs in about 10 to 50% of the adults with invasive amoebiasis. Symptoms which include abrupt onset of the high fever, tenderness in the upper part of the abdomen. Other symptoms might be anorexia, nausea, vomiting, fatigue, and weight loss. Some patients may suffer from mild jaundice because it is affecting the liver. Hepatomegaly, which is enlargement of the liver, is also one of the symptoms of hepatic amoebiasis. Amoebic liver abscess and its complications are responsible for about 40% of the death. Secondary bacterial infection may also result. Let us talk about the metastatic lesions in other organs, having talked about the metastatic lesions in spleen, liver, etc. earlier on. The pulmonary amoebiasis is a complication of amoebic liver abscess. However, in rare cases, the trophozoites may gain entry directly into the lungs from the gut via the portal circulation into the pulmonary capillaries, causing primary pulmonary amoebiasis. It occurs only in 10% of the patients with liver amoebic abscesses, and the mortality rate is 15 to 20%. Severe pleuritic chest pain extending up to the right shoulder, dyspnea, and non-productive cough are some of the symptoms of pulmonary amoebiasis. After talking about liver abscess, there are other metastatic lesions which also occur. For example, cerebral amoebiasis. It's a rare complication which has been documented only in about 0.65 to 4.67% of the patients with liver amoebic abscess. It is caused when the trophozoite gain entry in the brain. A small abscess is formed in the cerebral hemisphere and the patient may die within 12 to 72 hours because of the cerebellum is infected. Cerebral amoebiasis has a mortality of about 90%. Splenic abscess, it is yet another metastatic lesion. The trophozoites are able to gain access to the spleen, found in association with the liver abscess. Cutaneous amoebiasis is yet another metastatic lesion, less frequently encountered, of course. It can be caused due to perforation of amoebic abscess or of surgical wound infection with the entamoeba. No vaccine has so far been developed for amoebiasis in humans. There are three antigens, however, which are being tried out in experimental animals. These are recombinant 170 kD galactose, which is N-acetyl D-galactosamine, a 52 kD recombinant fusion protein, and a 260 kD antigen. Host immunity has also been reported in amoebiasis, and this host immunity can either be non-immune defense mechanism or it can be specific immunity. Let us take each one of them one by one. Non-immune defense mechanism. Non-immune defense mechanism means that immunity can be because of the gastric acid barrier, which kills the amoebic trophozoite, or it could be because of the rapid intestinal transit that reduces the time for the amoebae to colonize the intestinal mucosa, or because of the colonic mucin, which inhibits the amoebic adherence to the intestinal epithelial cells. Talking of the specific immunity now, this can be either humoral immunity or cell-mediated immunity. Humoral immunity. Humoral antibodies in primary infection appears to be responsible for elimination of the amoebae from the intestine and protection of the host from the amoebae against infection and causing further damage to the host. As has already been discussed earlier, during the process of amoebiasis, trophozoites are continually exposed to the lytic action of the complement present in the serum. The amoebae protect themselves from the lytic action of the complement because of the activities of the amoebic lectin present on its surface. The amoebic leptin binds to the C8 and C9 components of the complement, thus inhibiting their assembly and in the process preventing the lysis of the amoebae. Although all classes of immunoglobulins are produced, but IgG antibodies are most predominant during invasive amoebiasis. Now, what is cell-mediated immunity? It's a response which is characterized by the production of lymphokines, including interferon G, which induces the killing of trophozoites by macrophages. CD8 plus lymphocytes also play an important role in killing the trophozoites. Cell-mediated immunity plays an important role in limiting the extent of invasive amoebic disease and protects the host from recurrence of amoebic disease. Now let us talk a little bit about the genome. E. histolytica genome was the first human amoeba to have been sequenced. The strain was HM11MSS, assembled and automatically annotated in 2005, which was reassembled and re-annotated in 2010. It was found that E. histolytica contains 20 million base pair genome assembly having 8,160 predicted genes. Novel transposable elements have been mapped and characterized. The major group of transposable elements in Entamoeba histolytica are non-LTR retrotransposons. These have been divided in three families called EH lines, long interspersed elements, and short non-autonomous elements. Together, they account for about 6% of the Entamoeba histolytica genome. EH line 1 encode an endonuclease protein which have similarity with bacterial restriction endonuclease. This indicates that the transposable elements in Entamoeba histolytica have been acquired from prokaryotes by horizontal gene transfer. These studies on the presence of genes and the potential for novel metabolic pathways in E. histolytica may allow the development of new chemotherapeutic agents. Now let us talk a little bit about the epidemiology. It is estimated that about 90% of the world's population is asymptomatic. Only 10% may produce clinical symptoms. About 50 million cases of invasive amoebiasis are reported every year. About 100,000, that is roughly about 10% of the infected people, in them deaths have been reported annually. Amoebiasis is considered as the third deadly disease. The other two being, of course, malaria and schistosomiasis. Although the prevalence of Entamoeba histolytica is higher in males, more serious and fatal complications have been found, however, in women and children. Amoebiasis is a major health problem in Africa, China, Latin America, and Southeast Asia, including the Indian subcontinent. The prevalence of infection is about 50% in areas of Central and South America, Africa, and Asia, which we have already done, if you recall, in our earlier module. Transmission. How are these Entamoeba histolytica transmitted? Recall, students, we have done it in our previous module also, that fruits, raw vegetables, food contaminated by feces containing quadrinucleate cyst are the main methods of transmission for Entamoeba histolytica. Contaminated water, unhygienic habits, mechanical vectors like houseflies and cockroaches, they are yet another source for the transmission. Houseflies may act as carrier of the cyst from feces to unprotected food and water. The droppings of cockroaches too have been found to contain mature cysts, and thus they also serve as a source of infection. Sexual transmission of Entamoeba histolytica has been reported in about 20 to 30% of homosexuals. Now talking about the diagnosis. How can you diagnose Entamoeba histolytica infection? Yes, the foremost is fresh stool examination, because we have said that they are found in the mucosa and submucosa of the large intestine. Observation of fresh stool containing mucus and blood under the microscope is one of the easiest method for diagnosis. Wet mount of stool of trophozoites can be studied in saline preparation, while cysts, they show morphological details in iodine preparation. Stool should be examined as soon as possible. Trophozoites cannot survive for long outside the body of the host, so examination has to be done fast. Stool smears can also be examined after permanent staining by iron hematoxylin or by trichogrom or periodic acid shift, that is PAS stains. Internal structure of trophozoites are best studied by PAS staining. Trichrome stain is useful to study internal structure of both trophozoites as well as cyst. Talking about the diagnosis, we have said that stool examination is one of the easiest methods of diagnosis. Some other methods for diagnosis are sigmoidoscopy or colonoscopy, that is biopsy of the ulcer, which can be examined by histopathological slides of histopathological studies. Then serology, which is detection of antibodies to Entamoeba in serum, which has been found to be useful in invasive amoebiasis, and serological tests include enzyme-linked immunoassay, which is ELISA, indirect hemoagglutination assay, which is IHA, cellulose acetate precipitin, DNA probes and PCR, PCR is polymerase chain reaction, have also been used to detect Entamoeba histolytica in stool and liver aspirates. Exposing some stool to a strip of paper coated with antibodies can also be used as a diagnostic tool. The parasite in them will stick to the antibodies on the paper. What possible treatment can we give to persons who have Entamoeba histolytica or who are suffering from amoebiasis. Metronidazole is given to the patients with symptomatic intestinal and extra-intestinal amoebiasis. Tinidazole and ornidazole are believed to be similar in action to metronidazole. Paromycin is given for treating amoebiasis when metronidazole is not effective. Antibacterial drugs may be necessary in patients with complicated intestinal amoebiasis to prevent secondary infection of intestinal ulceration. However, one should not take any of these medicines because self-treatment is very bad. One should always consult a doctor. Prophylaxis. Prophylaxis can be at individual level and at community level. Let us talk about the individual prophylaxis. We've already done this in our earlier module, where we talked about the morphology and life cycle of Entamoeba histolytica. Cleanliness and personal hygiene is very important to prevent such infections. Wash hands after defecation and before eating. Drink safe and uncontaminated water. Boiling water is a good practice, but there is a caution. Cysts of Entamoeba are resistant to the chlorine concentration which is used to disinfect water. Eat properly washed vegetables and fruits. These can be treated with acetic acid or vinegar for 15 minutes before eating. Vinegar is easily available in houses. Sanitary disposal of feces should be taken care of. Care should be taken to keep the mechanical carriers like houseflies and cockroaches under control. Keep the food stock covered. Talking about the community prophylaxis, improvement of general sanitary disposal of feces, prevention of water supplies from getting contaminated by feces, and early detection and treatment of asymptomatic carriers are some of the prophylactic measures which can be taken at the community level. So students, let us now summarize what we have learned in this module. We know that the Entamoeba infection can either be non-invasive or invasive. We also know that Entamoeba histolytica is the etiological agent of amoebiasis, acute dysentery, chronic amoebic dysentery, and the invasive form may be the cause for liver abscess. Not only liver abscess, splenic abscess, pulmonary and cerebral amoebiasis. The parasite may be a cause for ulcers in the large intestine. Certain strains of Entamoeba histolytica can be more virulent than the others. So far, no vaccine has been developed for amoebiasis in humans. Although humans do show immunity to amoebiasis, which can either be specific immunity or non-immune defense mechanism. Fresh stool examination, sigmoidoscopy or colonoscopy, serological tests, DNA probes, and PCR. PCR means polymerase chain reaction are some of the methods used for the diagnosis of Entamoeba histolytica in humans. The preventive measures that can be taken for Entamoeba histolytica are cleanliness and personal hygiene, drinking safe and uncontaminated water, eating properly washed vegetables and fruit, and proper sanitary disposal of feces. Care should also be taken to keep the mechanical carriers like the houseflies and cockroaches under control. Thank you, students.